Imagine a tumor as a heavily fortified castle. For decades now, we've been sending in our absolute best soldiers.
Right, our T cells.
Exactly, our T cells. But we just watched them get exhausted, confused, and, well, ultimately blocked at the gates. We've tried training them, arming them with new weapons, but those castle walls remain incredibly stubborn.
And, it actually goes beyond just a physical barricade. Solid tumors actively secrete these chemical signals that create a highly immunosuppressive microenvironment.
Which is basically an active suppression zone, right?
Yeah, it effectively puts our circulating T cells to sleep before they can even draw their weapons. That's really why CAR T-cell therapy, which is, practically a miracle for liquid blood cancers like leukemia.
Yeah, it's been amazing for that.
It has. But it often hits a total brick wall when confronted with a solid mass, like breast, lung, or ovarian cancer.
So if the highly trained soldiers are just getting put to sleep at the gates, maybe we need a totally different strategy here. What if instead of soldiers, we sent in a limitless swarm of heavy-duty garbage disposals?
To literally eat the castle from the inside out.
Yes. Which brings us to our topic today, the macrophage. Macrophages are basically the body's natural vacuum cleaners, right?
That's a great way to put it. They wander into our tissues, clear out dead cellular debris, and engulf pathogens. So using them to eat solid tumors sounds perfect on paper.
Because they naturally want to penetrate tissues anyway.
Exactly. But biology throws a massive wrench into this plan when it comes to manufacturing them at scale.
Right, because if you're listening to this deep dive and wondering, why don't we just grow 1 billion macrophages in a lab and inject them? It comes down to this really frustrating concept called terminal differentiation.
Yeah, terminal differentiation is the bottleneck. To use a visual, think of human development. A mature macrophage is like a fully grown adult.
It's reached its final form.
Right. Once a cell commits to becoming a mature macrophage, it effectively, permanently shuts down its ability to divide. It will never split into 2 cells again.
Wow. So if you extract macrophages from a patient to engineer them, you just cannot expand their numbers in a petri dish.
Not at all. Whatever number you harvest is the absolute maximum number you have to work with.
Which is a nightmare if you're trying to dose a patient with the billions of cells required to shrink a tumor. You have no way to draft or train new recruits.
And to make matters worse, when we do try to inject those mature macrophages back into a patient, they don't even go to the tumor.
Wait, really? Where do they go?
They get sequestered. You see, mature macrophages are physically large and highly adhesive.
Oh, so they're sticky.
Very sticky. When you infuse them into a patient's bloodstream, they hit the incredibly fine microcapillary beds of the lungs, and they just get physically wedged there.
That sounds problematic.
It is, or they get filtered out by the liver and spleen. So an incredibly tiny fraction actually navigates to the tumor site.
So to summarize the problem, we have a therapy that won't multiply, and when you inject it, it just gets trapped in the lungs. It feels like a total dead end.
It did feel like that for a long time.
But today, we're doing a highly detailed deep dive into the absolute cutting edge of cellular immunotherapy. We're unpacking a groundbreaking paper from the journal Cell that completely rewrites this rulebook.
It really does. The researchers found a way to bypass the mature macrophage entirely.
Right, by looking at a precursor cell, which is called a granulocyte-monocyte progenitor, or GMP for short.
Yeah, so instead of trying to force an adult to clone itself, the researchers went backward up the family tree, so to speak.
Kind of looking at the stem cell hierarchy.
Exactly. In the hierarchy of our blood system, you have hematopoietic stem cells at the very top, like the trunk of the tree.
Yeah.
They can turn into anything.
Okay, I'm visualizing the tree.
As cells divide, they move out into the branches, committing to specific paths. A GMP is a branch that is committed to becoming either a granulocyte, like a neutrophil, or a monocyte.
And that monocyte eventually matures into a macrophage.
Precisely.
So if the mature macrophage is the fully armored knight who can't clone himself, the GMP is like the military boot camp. It's the factory.
That's a perfect analogy.
But hold on, if I remember my biology basics, textbooks describe these intermediate progenitors as transient amplifiers.
Yep. That's the standard dogma.
Right. They're supposed to do a few quick rounds of division and then immediately grow up into mature cells. So how do you stop them from maturing so you can actually stockpile them?
Well, you're hitting on the central assumption this paper challenges. The belief was always that a committed progenitor cannot be kept in a prolonged state of self-renewal.
Like the biological clock is always ticking.
Exactly. But these researchers hypothesized that if they could discover the precise chemical signals the cell uses to mature, They could block them.
So they wanted to halt biological time, freezing the GMPs in a state where they do nothing but copy themselves without ever losing that core stem-like identity.
That was the goal, yeah.
Okay, let's do a quick recap for the listener before we move on. We've gone from CAR-T failing at the castle gates, to macrophages getting stuck in the lungs, to now needing to find a chemical pause button for these GMP factories.
That's exactly where we are.
So think about this if you're listening. If you were a scientist, what kind of chemical signals do you think you'd need to freeze a cell in its youth? How do you even begin to find a pause button for a living cell?
Well, you start with a massive screening process using mouse cells. They literally tested hundreds of different compounds and growth factors.
Just trial and error.
Pretty much. Looking for anything that would keep the cells dividing without maturing, and they eventually hit the jackpot with a 3-part chemical recipe.
Right, and they named it SCF2I. Let's break down the chemistry of that cocktail. What is it actually doing to the cell?
Okay, so the first ingredient is SCF, which stands for stem cell factor. This is a natural protein that simply provides a baseline survival signal.
So it just keeps them from dying.
Right. Without it, the cell undergoes apoptosis-programmed cell death. The magic really happens with the 2I, which stands for 2 small molecule inhibitors.
Got it. What's the first inhibitor?
A drug called GDC-0879. This is a BRAF inhibitor.
Wait, I've heard of BRAF in the context of melanoma treatments. It's a protein that usually tells the cell to grow, right?
Yeah, it's a key node in the MAP-Kirk signaling pathway. In many cancers, a mutated BRAF is stuck in the on position, driving uncontrolled tumor growth.
So why inhibit it if we want the cells to grow?
Because in these specific myeloid progenitor cells, that pathway actually drives differentiation. It pushes the cell to mature.
Oh, wow. So by pharmacologically blocking BRAF, they're severing the chemical signal that tells the cell to grow up.
Exactly. It's incredibly elegant.
It really is. And the second inhibitor in the 2I cocktail?
That's a compound called SKL-2001. When they combined SCF, the BRAF inhibitor, and SKL-2001, the mouse GMPs did something unprecedented in cellular biology.
What happened?
They entered log phase expansion for over 50 days.
Log phase expansion. Let's translate that into practical terms for the listener. How many cells were we talking about here?
They multiplied by 10 to the 13th power.
Wait, 10 trillion cells from a tiny microscopic starting batch.
Yep. To visualize it, if you didn't have to keep splitting them into new flasks, and they didn't, consume all the nutrients in the lab.
Right, theoretically speaking.
You'd have enough cellular mass to start filling a swimming pool.
That is insane.
And the crucial part, after 50 days of this aggressive cloning, they sequenced the cells. They hadn't drifted.
Meaning they maintained their exact biological identity as granulocyte-monocyte progenitors?
Exactly. The numbers game was completely solved.
So you have an infinite off-the-shelf supply of boot camp factories, but, and there's always a but, in early stage science, mice are not humans.
No, they are not.
When they poured this magical mouse cocktail onto human stem cells, what happened?
The human cells barely proliferated at all. The translation completely failed.
Oh man, so what do you do at that point? You know the concept works in mammals, but the human biology is rejecting the specific chemical key. Do you just start mixing new chemicals blindly?
Not blindly, no. You isolate the weak link. Through elimination, they discovered that the first 2 ingredients, the stem cell factor and the BRAF inhibitor, worked perfectly fine in human cells.
So it was a third ingredient.
Right. The failure was SKL-2001. Its chemical activity just wasn't potent enough for the human cellular machinery. So the chemists had to build a better key.
How did they do that?
They synthesized and screened over 120 structural analogs. Essentially, they were tweaking the molecule's shape slightly over and over until they found one that clicked into the human receptors perfectly.
And what was the winning molecule?
They named it TN-230. They swapped that into the recipe, added 2 more natural human growth factors called IL-3 and GCSF just to keep the cells happy, and created the human cocktail.
Which they called SCF2I-IG.
Exactly. And it worked beautifully. The human GMPs expanded continuously, maintaining their progenitor state without a single genetic mutation.
But the story of that third ingredient gets even wilder. Because for years, the scientific community thought they knew exactly how that original drug, SKL-2001, worked. Textbooks claimed it was a modulator of the Wnt beta-catenin pathway.
Yeah, that was the accepted dogma. The Wnt pathway is this classic cell signaling route involved in stem cell renewal. Everyone just assumed SKL-2001 was turning that pathway on.
But the researchers in this paper ran a brilliant experiment to verify it, didn't they?
They did. They took a genetically modified mouse where the beta-catenin gene was entirely deleted. The pathway was physically gone.
So if the drug relies on that pathway, pouring the drug on those modified cells should do absolutely nothing.
Right, but the cells expanded perfectly.
Wow. So the Wen pathway had absolutely nothing to do with it. The entire field was wrong.
Completely wrong.
Yeah.
SKL 2001, and by extension the new human drug TN230, was hitting an entirely unknown, invisible target inside the cell.
So how on earth do you find a single mystery protein in the soup of a living cell? I mean, you can't just guess.
No, you have to go fishing at the molecular level. They used a technique called click chemistry.
Oh, click chemistry. How does that apply here?
They took a highly potent version of their chemical and synthesized a tiny molecular handle onto it, an alkyne tag. Think of it as a specific carbon chemical bond that acts like Velcro.
Okay, so you have a chemical with a strip of molecular Velcro attached to it.
Exactly. They drop this probe into the ground-up contents of the cell. The chemical naturally binds to its mystery target.
And then?
Then they introduce another molecule, a fluorescent bead, that covalently clicks onto that alkene Velcro tag.
Oh, I see.
They use that bead like a hook to pull the chemical and whatever secret protein is stuck to it right out of the mixture.
The ultimate molecular fishing trip. That is so cool. And the protein they pulled out of the cellular soup?
An enzyme called myeloperoxidase, or MPO.
Great. Myeloperoxidase, I know that enzyme. That's the stuff inside our white blood cells that literally generates reactive oxygen species, right? Basically, cellular bleach to destroy engulfed bacteria. What is an antibacterial bleach maker doing controlling cell division?
That discovery right there is what makes this paper a landmark. MPO is famous for killing bacteria in mature cells. But they discovered that in young progenitor cells, MPO has a moonlight job.
A moonlight job, like a side hustle.
Yeah, exactly. The low levels of reactive oxygen species it produces actually act as an internal stress signal. It is a biological clock or a brake on the cell cycle.
Oh, so when ROS levels hit a certain threshold, the cell stops dividing and starts maturing.
Precisely. So by pharmacologically inhibiting MPO with their custom chemical, they are lowering the reactive oxygen species, which releases the brake.
Tricking the cell into thinking it's not time to grow up yet.
Right. And the paper notes that pouring this chemical on the cells phenocopies a genetic knockout.
Meaning using the drug achieves the exact same physical result as using a gene editing tool to literally cut the MPO gene out of the cell's DNA.
Yes. And this is a massive safety advantage for human therapy. We get infinite scalable manufacturing without ever having to permanently mutate the cell's genome.
We just pause it with a chemical, and when we wash the chemical away, the cells resume their normal, healthy development.
Exactly.
This is a great moment to quickly recap and tie these concepts together. If you're listening and marveling at the complexity here, think about this. We've solved the manufacturing bottleneck.
Right. SCF2I for mice, SCF2IIG for humans.
We discovered that a drug we thought did one thing actually hits a hidden brake called MPO. And by releasing that brake, we can grow trillions of human immune progenitors in a lab.
So consider the implications here for a second. If we can pause biological aging in one type of immune cell, what other cells might have hidden brakes we just haven't found yet?
It's a crazy thought. But, having a swimming pool of young cells is only half the battle. If we take these naive recruits and put them into a living body, do they actually know how to fight?
That was the next major hurdle. They had to prove in vivo, so in a living organism, that these lab-expanded GMPs could mature into functional immune cells.
And what model did they use for that?
They use a really fascinating baseline model, the CGD mouse.
CGD, that stands for chronic granulomatous disease, right? What is the specific defect in these mice?
So CGD is a genetic immunodeficiency. The mice's natural phagocytes, their eating cells, are broken. They cannot produce the oxidative burst needed to kill the bacteria they swallow.
Oh, so they just get overwhelmed.
Yeah, they succumb to incredibly common, simple bacterial infections like Staphylococcus aureus or B. cepacia. They develop severe liver abscesses and die.
Okay, so the researchers take these sick, immunodeficient mice, and they inject the lab-grown paused GMPs into their bloodstream. What happens?
Without any further instruction, the injected GMPs circulated, found their way into the bone marrow, and naturally matured into healthy, functioning macrophages and granulocytes.
Just on their own.
Just on their own. They went to work, they completely cleared the bacterial liver abscesses and saved the mice's lives.
Wow.
Yeah, this proved definitively That taking the break off in the lab didn't break the cell. They retained their full, natural biological potential once placed back in a body.
Okay, so the recruits can fight standard bacterial enemies, but the mission here is cancer. We need to engineer them. We need to give them a map to the tumor.
Right, we have to arm them.
But hold on, you mentioned earlier that mature macrophages destroy pathogens. If we try to use a lentivirus to insert a new cancer-tracking gene into a macrophage, wouldn't the cell just recognize the virus as an invader and destroy it?
You nailed it. That is exactly why engineering mature macrophages has been a historical nightmare.
Because they're doing their job.
Exactly. Mature macrophages are packed with innate viral sensors. When you try to insert a gene using a viral vector, those sensors trip, and the cell often undergoes apoptosis to protect the body.
It basically self-destructs.
Yep. But GMPs are young. They haven't fully developed those mature viral sensor networks yet. Because they are actively dividing progenitors, they happily accept genetic edits.
So they were basically a blank canvas.
Exactly. The researchers easily modified them using lentivirus and CRISPR-Cas9 without triggering cell death. They armed the GMPs with chimeric antigen receptors, or CARs.
So they gave them a GPS for cancer. I know they tested this on a blood cancer model first, engineering human GMPs with an anti-CD19 CAR to hunt down B-cell acute lymphoblastic leukemia in mice, which showed durable disease control.
Yes, that was a huge success.
But the holy grail, the whole reason we're doing this depth dive, is the solid tumor.
Right. For the solid tumor, they used an aggressive ovarian cancer model called ESCO-V3, which overexpresses a marker called HER2.
Okay, so they engineered the GMPs with an anti-HER2 CAR and injected them.
They did.
And the imaging data from this experiment is simply stunning. Because remember how we said mature macrophages just get physically wedged in the lungs and glow brightly in the chest on a scan?
Right, the sequestration problem.
Well, the researchers used a dual luciferase tracking system. The solid tumor glowed one color, and the engineered GMPs glowed another.
And what did the heat map show?
It showed that these young, nimble GMPs completely bypassed the trap of the lungs. They homed in on the solid tumor and densely packed themselves inside the tumor tissue. They literally swarmed the castle.
It's incredible. And the mechanism behind this swarm is a total paradigm shift in therapy.
How so?
When you inject a mature cell, it's a one-and-done event. But by injecting a progenitor cell, you leverage the body's natural biology. The GMPs actually seed the host's bone marrow and create a continuous wave of what we call donor-derived myelopoiesis.
Let's translate that. Myelopoiesis is the creation of new myeloid cells. So you're saying the therapy sets up like a permanent forward operating base inside the bone marrow.
Precisely. It acts as a living, self-replenishing drug. As the engineered macrophages inside the tumor eventually die off from exhaustion, the bone marrow base camp constantly produces and deploys fresh CAR-engineered macrophages to replace them.
So it is a relentless, continuous assault on the tumor microenvironment. It's brilliant. Okay, so quick recap for you listening. GMPs cure bacterial infections in CGD mice. They're easily engineered with CARs. And they successfully infiltrate and destroy solid tumors and leukemia from a base in the bone marrow.
It sounds like case closed, right?
Right. But cancer is insidious. Even a swarming army of macrophages might not be enough to eradicate a massive solid tumor alone. So we have to ask, we've solved the numbers game and the tracking game, but how do we get the rest of the patient's natural immune system to wake up and join the fight?
And that question brings us to what I honestly consider the absolute masterstroke of this entire paper.
The masterstroke, let's hear it.
Up until this point, the engineered human GMPs worked beautifully in immunodeficient mice. But in a real clinical scenario, especially if we want to use an off-the-shelf therapy where the therapeutic cells come from a healthy donor rather than the patient themselves, we run headfirst into a massive biological wall.
MHC mismatch.
Exactly.
MHC stands for major histocompatibility complex. I like to think of it as cellular passport control.
That's a perfect analogy. Every cell in your body displays an MHC molecule that acts as a password, telling your immune system, hey, I belong here.
Right, and if a donor cell doesn't have the right password, the host's immune system usually just attacks it.
Exactly. But even if we overcome rejection, there is a severe communication breakdown. Traditional CAR macrophages are great at eating the tumor, a process called cis-phagocytosis. The engineered cell directly binds and engulfs the cancer cell.
But once they eat the tumor, they just keep the evidence to themselves.
Yeah. Because they are donor cells with foreign passports, they cannot effectively interact with the host patient's T cells. They can't display the broken down pieces of the tumor to the host's adaptive immune system.
It's like a lone wolf vigilante taking down a criminal in a back alley. They do the job, but the local police department has no idea a crime ring even exists, so they don't send the SWAT team.
That's exactly it. The broader immune system remains completely blind to the cancer.
So how did they fix this communication blackout?
The researchers made an ingenious modification to the molecular architecture of the CAR itself. They created the CAR-FC.
Okay, CAR-FC. What does the FC stand for?
They took a standard CAR receptor and inserted a very specific molecular domain, an immunoglobulin G1 or IgG1 FC domain, into the portion of the receptor that protrudes outside the cell.
Break down the FCD domain for us. What does that tiny molecular tail actually do?
The FCC domain is essentially the tail end of a natural antibody.
Yeah.
The host patient's own local unengineered macrophages have specific receptors on their surface called Fc gamma receptors.
And what do those do?
These receptors are specifically designed to grab onto Fc domains. They act like biological tow cables.
Oh, so when the engineered donor cell binds to a tumor cell, that protruding Fc tail acts as a universal beacon.
Exactly. It triggers what is called transphagocytosis.
Transphagocytosis, meaning across different cells.
Right. The host's local macrophages see this Fc beacon, latch onto it with their tow cables, get pulled aggressively into the tumor microenvironment, and are literally forced to start eating the target right alongside the donor cell.
To use our analogy again, the CAR-F isn't just a vigilante. It is a vigilante who shoots a massive flare gun into the sky. It forcibly drags the local cops, the host's own immune cells, to the scene of the crime, makes them take evidence, and puts out an all-points bulletin to the T-cell SWAT team.
That is exactly what happens. And because the host's own macrophages are now eating the tumor, the passport problem is completely solved.
Because the host macrophages possess the correct MHC passwords.
Right. They can travel to the local lymph nodes, present the tumor antigens, and activate the host's endogenous OTT T cell.
They basically trick the patient's body into vaccinating itself against its own tumor.
It's brilliant.
But did they prove this works in a fully intact immune system? I mean, mismatched models.
They did. They tested this CAR-F design in fully immunocompetent allogeneic mouse models, meaning models with a completely mismatched immune system.
What types of cancer?
They used aggressive A20 lymphoma and 4T1 breast carcinoma, and the results were undeniable.
I can imagine.
The CRFC triggered a massive infiltration of activated host-derived T cells deep into the tumors. It completely bridged the gap between the innate immunity of the donor cells and the adaptive immunity of the host,
leading to improved survival.
Drastically improved survival over standard CR designs.
It is a stunning piece of biological engineering. So pulling all these threads together for you, listener, we started with the problem of mature macrophages being impossible to manufacture and getting stuck in the lungs.
Which they solved by going upstream to GMPs.
Right. They found that a mystery drug actually blocks the MPO enzyme, releasing a biological break and allowing us to grow trillions of these cells in a dish.
And because they're young, they can be easily armed with CARs and will swarm a solid tumor from a forward operating base in the bone marrow.
And finally, by adding an IgG1 flare gun to the CR, they force the patient's own immune system to wake up and finish the job.
It really represents a fundamental shift in how we view cellular medicine. We are moving away from seeing cell therapies as static terminal drugs and instead designing them as living, dynamic systems that actively manipulate and recruit the host's own biology.
Which brings us to one final, incredibly counterintuitive insight from the paper's discussion. If you follow CAR-T therapy, you know the ultimate goal is usually persistence.
Right. We usually want the engineered cells to live forever in the patient's body to prevent a relapse.
But because these GMPs come from a healthy donor, the host's immune system will eventually reject them after a few weeks, even with the stay-off-a-C trick.
You'd think that's a bad thing. But surprisingly, the paper argues this transient persistence is actually a massive advantage.
How is getting rejected an advantage?
It completely reframes safety. Think about it. If an engineered immune cell lives forever, you have to be very careful about what target you give it.
Why?
If you target a marker that is on the tumor, but also present in small amounts on healthy lung or liver tissue, permanent cells will cause chronic, potentially fatal autoimmune damage.
Oh, I see. But because these GMPs have a strict expiration date, the rules change.
Precisely. Because we know the donor GMPs will be cleared from the body in a few weeks, we can safely target highly aggressive, widespread tumor markers that we wouldn't dare touch with a permanent therapy.
Wow. We can hit the cancer incredibly hard and fast, knowing the engineered cells will naturally die off and wash out of the system once the host's own T cells have been educated and have taken over the fight.
It completely flips the script on how we design living drugs.
It really does. Maybe the perfect therapy isn't a permanent resident that patrols the body forever. Maybe the perfect therapy is just a temporary teacher. You don't need the boot camp instructors to fight the entire war. You just need them in the trenches long enough to train the local army.
That's a profound way to look at it.
Well, if you are listening to this, I want you to really think about that flare gun concept, the CAR-FC. We are tricking a body into recognizing a threat it was previously blind to.
The applications could go way beyond just cancer.
Exactly. How else could we use that? Could we use temporary swarming immune cells to train the body to hunt down persistent viruses, or even reset the immune system in autoimmune diseases?
The possibilities are endless once you know how to build a factory.
Something to ponder as you go about your day. We'll catch you on the next deep dive.